And so what that does is it sets up a sort of feedback loop where humans push into these previously pristine areas. And because of that, both they and their livestock are coming in contact with species that humans have never been in contact with before. And by doing so, they're exposing themselves potentially to a range of new pathogens that our immune systems aren't set up to combat. And so by becoming potentially infected with these, either them or their livestock, this leads to livelihood impacts and also economic pressures. Overall, you find that in countries that are plagued by infectious diseases, the economies don't grow as well as they should, and so this leads to more of a push to gain more resources and push into areas that were previously pristine. One of the big players in this are the extractive industry. Whether that be mining or lumber, groups will come into these countries and push into previously pristine areas. And the first thing that they do is they build a road to these areas where they're extracting the minerals of the trees. And once you're out in that area, you have a large number of employees that you need to feed. So the easiest thing to do is to go off into the field and catch what's around you. Oftentimes you end up with extra food, and now you have a nice road that takes you right back to the capital city. So it leads to a movement of animals and the viruses that they can transmit. This is particularly risky in areas where there's shared or limited resources. So this is a photograph taken in Tanzania. You can see a young woman and her child. She's getting some water for herself and bathing at the same time as she's collecting water for dinner that evening. And she's also then allowing her herd to drink some water. What you can't see here, but if we were actually present here, I could take you around to the other side of the lake where there's a large number of diversity of footprints from wildlife. So in the evening hours or at sunrise or sunset, a large range of wildlife species also use this pool. So in a sense, this is actually literally a mixing pool of potential pathogens from all of these species. So I just want to take us back to this graph again. If we want to move towards a better approach of detection and identification of viruses early on, thereby increasing our opportunity for control, we need to move towards a more predictive approach. And to do that, what we believe you need to do is just look over the horizon a little bit and get a sense for what's out there and what has the potential to cross over into humans. And by doing this, you can get a sense for the various viruses that are there, and you can develop assays and be prepared for them should you see any odd illnesses arrive. A lot of people call this approach the One Health approach, the linking of animal health and wildlife health to human health. So by paying attention to what's going on in the wildlife, we can get a sense for what could be coming in humans. This is the approach taken by one of the main projects that I work on. It's funded by the United States Agency for International Development, and it's part of their Emerging Pandemics Threats program. The program is called PREDICT. They're not too creative on the names, but the main goal of it is to build a global early warning system for emerging diseases that move between wildlife and people. And the main goal then is to increase local capacities in global hotspots in order to detect viruses before they emerge in humans. So if you want to set up such a system, it seems like a daunting task. The first question is, where do you start? Perhaps the first question after that is, what do we test? By some estimates, there are about 9 million species on the planet. So we have to take a more strategic approach since we can't test everything. So as you might expect, we've developed a targeted strategy of surveillance of wildlife. And our strategy is to look at wildlife species that have enhanced risk for harboring viruses that cross over into humans. So you can see a graph here that shows the proportions of viruses shared by a variety of taxonomic groups between them and humans. And you can see that some taxonomic groups, in particular, bats, rodents, and non-human primates, have a very high proportion of viruses shared with humans. And this is notable given that all of these taxa of animals have about the same number of viruses that inhabit them. But these three groups share a much higher proportion of their viruses with humans. And we think that one of the reasons for that has to do with proximity, both genetic proximity as well as spatial or physical proximity. For instance, with non-human primates, as the name implies, we're quite closely related to them. In fact, the difference genetically between humans and chimpanzees is less than the distance between rats and mice. And so a virus that's adapted itself to infect a chimpanzee doesn't have very far to leap in order to infect humans. It's already adapted to a lot of the cells in the immune responses. With bats and with rodents, it appears to be more of the case of spatial proximity. Even in the US, you would say that rodents are ubiquitous. They're in just about every house, whether you be in the US, in Europe, or in the tropics. But especially in the tropics, there's much less in the way of barriers between these rodents and, for instance, the food. So in the evenings, the rodents will come out and eat off of the plates that the humans have finished eating off of. But also they can defecate and urinate into food or on the floor. Bats also are ubiquitous, and they've done quite well in both urban and semi-urban environments. And particularly in the tropics, their populations are quite large, and they're associated with a lot of human developments. You find them in the roofs or the ceilings of a lot of houses, and also associated with areas where people store food. So either through direct contacts with bats or contact with their droppings, they can share their viruses with humans. Also, if you think about it, you say that when maybe 10,000 years ago, you called us cavemen, and bats also like caves. So they had a lot of opportunity to interact with us prior to us figuring out how to make houses. So the next question is, now that we've identified what we would test, is where do we test? Clearly the world's a very big place, and we can't test everywhere. But as the map that I showed you earlier kind of implies, we've developed a strategic approach to where we sample as well. And so we've targeted the areas that we sample based upon the risk of emergence across the globe. So we've both taken into account all of the locations where previous emergence events have happened, but also modeling emergence and getting a sense for where in the world's emergence events are most likely to happen. In some of our earlier maps, we just overlaid factors like human population density with wildlife diversity. And since then, our models have become a little bit more complex and taken a lot more factors such as landscape changes and different land uses in those areas.
Question?
Yep.
It appears that Las Vegas and New York City are highly at risk. Why would that be?
So here's the thing. When you look at previous emergence events, you have reporting bias. So especially in the Northern Hemisphere for, say, the last 500 years, a lot of emergence events were happening across the globe. But those that were reported were mostly in the Northern Hemisphere. So on the first pass of this data, it looks like New York and France are, in those areas, are the highest risk. But it has to do a lot with reporting bias. And we fixed that in our newer models by developing an algorithm that goes through all of the peer-reviewed literature and scraping data off of that literature to find out the country of origin of the author. And that gives us sort of a background reporting activity level for all areas, and then we can adjust it based on that. I'll get into, this shows one of our newer models, and we've gotten down to a 1 kilometer grid. So we can really be strategic, even within the countries, as to the locations that we focus our surveillance efforts on. But models and data from computers don't give you all the information you need. It looks pretty good when you look at a country like Malaysia. You can say, oh, we definitely want to look over in the peninsula there and some of those other red areas. But when you get to a country like Rwanda, the model paints the whole thing red. So we've had to have our teams on the ground pull in actual real-world data. And the people that we hire all live in the country, so they're very familiar with the systems and the cultures there. And so they can pull in a wealth of data that can help us to direct our surveillance into areas that make more sense. So information like where's the wildlife trade going on? In what regions is hunting more likely to occur? ports of entry and exit, land use change, and also extractive industry. And so we've overlaid that into our maps to make everything far more strategic and targeted, which makes the surveillance more efficient and more likely to detect these emergence events. So overall, both geographically and with the species that we select, we've developed this strategic approach, a targeted approach. But on top of that, this would still be too big of a project. On top of that, we've also overlaid risk. So for instance, we don't want to sample a bat that's 400 miles from the nearest human, because clearly that bat isn't, at this point, a big risk to humans. So we sample wildlife along high-risk disease transmission pathways. So as I previously alluded to, we're looking at animals involved in the wildlife trade or animals being hunted. But then for free-range caught animals, we look at free-range wildlife that are in areas where disease emergence is promoted by human activities, such as landscape or land use change, sharing of limited resources, or incursion of humans into previously pristine areas. But additionally, we pay attention to animal die-offs and outbreaks, as these oftentimes mark the precursor of human infections. It makes a lot of sense if you think about it. If a large number of animals suddenly become infected with something, there's a lot more virus in the environment, and there's a heightened chance for humans to become infected. So these are some images of our teams in the field collecting samples from bushmeat hunters. And working with the communities on this is very essential. If you just show up in a village and ask for animals, as maybe some of our team members might have done at the beginning, you find that you get a lot of resistance. Early on, there's fear associated with collection of samples from bushmeat. In a lot of countries, hunting is actually illegal. So the fear is that you'll take their animals or you'll take their food. But over time, we've developed trust with the communities. And so that we're able to collect samples, some very valuable samples to test. But additionally, that interaction with the community gives us an opportunity to help educate them and reduce their risk of becoming infected. Because we don't want humans as sentinels. The purpose isn't for scientific discovery. The purpose is to reduce the risk of transmission of viruses into humans and maybe not stop outbreaks, but stop pandemics. So most of these pictures are taken in Central Africa, although I believe the one on the bottom right is from Asia. And these are just a couple images from our free-range wildlife collection. And one thing I'd like to point out about our free-range wildlife collections is that all of our collections are catch and release. At the beginning of this project, we decided that we didn't want to scour the globe in all of these interesting tropical places and kill off a bunch of cool wildlife. So we developed our approach such that we could collect samples and then release the animals. A lot of these involved blood samples or swabs, things that you can take without cutting an animal open. With the larger non-human primates, we had to develop different approaches. Capturing large primates, as you could expect, is quite challenging and disruptive to the populations. So in most of the countries where we work, we've partnered with wildlife sanctuaries. And they receive a lot of non-human primates that are either sick or have been injured, hit by a car, or have been confiscated. And so as they're brought into the sanctuaries, they collect samples for us, which we can then send on for testing. Although we have adapted a couple of interesting experimental approaches to try to collect samples from non-human primates. I mean, clearly you can collect feces, which isn't the best job, but you can then get samples from primates and later on go back and sequence them and figure out what primates they're from. One of the approaches we've tried out is you can toss around bundles of twine, just balls of twine. And for some reason, a lot of non-human primates find them very interesting. So they pick them up and chew on them for a while. And then they figure they're not food or they lose interest, so they spit them out. So you can take those and then take a saliva sample from them and test them for pathogens. Sometimes you need to be a little bit innovative with these animals. We've also developed some approaches to allow the hunters and the individuals at risk to help us collect samples. We can't be in all of these locations all of the time, so this shows one of our earlier attempts where we had the hunter collect little spots of blood onto filter paper after they caught their prey. And if you allow that to dry at room temperature, it's actually fairly stable for a fair amount of time, and so we can send people to pick them up occasionally. Doing this, especially in Central Africa, but also in other places, required us to make an ID guide for the Central African hunters. And this is important because village to village, you oftentimes have entirely different words for these animals, and you need to get some sense for what you're collecting. So in addition to looking at wildlife and looking just over the horizon, we also want to look at these individuals who are at that high-risk interface. So we also take samples from humans as well. And we use the same risk-based approach for them. Clearly, you don't want to collect a sample from someone in a major city who's never seen any wildlife in his life. Instead, we sample people that are at a high level of sustained and direct contact with the pathogens in animals. So wet market employees, hunters, people living in transition areas, and also people who have reported contact with wildlife, especially people who have reported contact with wildlife that's caused any sort of injury, like scratches or bites. But also we pay attention to acute illnesses of unknown origin. And what I mean by this is any illness that they've tested for the usual suspects. So for instance, in many of these areas you would test for dengue or malaria. And for those that came back negative, we'll take those samples and then try to identify what's going on and what the etiologic agent is. In particular, we pay attention to some of the more unusual or atypical presentations of diseases, and also, of course, viral hemorrhagic fever-like symptoms. So people who are bleeding out have symptoms like Ebola. These are just some pictures of some of the individuals that we work with to collect samples. An African hunter on the bottom left, as well as a bunch of market workers from Guangzhou on the top right. And as much as possible, we try to collect samples longitudinally as well, so that we don't just get a snapshot of time to see what might be crossing over, but we can get a sense temporally how things are crossing over, and we can see if there are any patterns to that cycle. The only person I've ever seen who smiles while he's getting his blood drawn there. He probably knew his picture was being taken. So we've been able to collect a really large number of samples from humans and also from a broad range of wildlife. And the types of samples that we collect are listed here. As I mentioned, we collect a large number of swabs, as well as urine and feces, if those are available. Oftentimes when you're handling an animal, it defecates, so it makes it really easy to collect those. And in some cases, especially when dealing with bushmeat, we'll have tissue samples as well.